our next chapter is regarding cell cycle and cell division. Okay, so coming to the topic of cell cycle and cell division, all we are aware of that even the largest start their life from a single cell. So we have the largest thing. So coming to the cell cycle is referred to as the sequential, coordinated and genetically control events. So control events are called cell cycle. So it is been happening in a cyclical way. You may wonder how a single cell then goes on to from such a large organism. So growth and reproduction are the characteristics of the cell. Coming to the growth and reproduction are the inclusive features for single celled organisms. So growth and reproduction are for single celled organisms. They both are inclusive in nature. So coming to the next thing, growth and reproduction for higher plants and animals are exclusive. So the same thing for higher plants and animals are exclusive. Okay, all cells reproduce by dividing into two. So each parental cell gives rise to two daughter cells each time. So they divide. The newly formed daughter cells can themselves grow and divide. So gives rise to a new cell population, and that is formed by growth and division. So growth and division of single parent cell and its progeny. In other words, such cycles of growth and division allow a single cell to form a structure consists of millions of cells. So coming to the cell cycle, it is the most very important process in all the living organisms. So during the division of cell, DNA undergo replication, cell growth takes place. All these processes, that is cell division, DNA replication and cell growth. Hence, they have to take place in a coordinated way to ensure the correct division and formation of progeny cells containing intact genomes. The sequence of events by which cells duplicates its genome, synthesis the other constituents of the cell, are eventually divided into two daughter cells is termed as cell cycle. So although the cell growth in terms of cytoplasmic increase is a continuous process, DNA synthesis occurs only at one specific stage in the cell cycle. The replicated chromosomes are then distributed to daughter nuclei by a complex series of events during cell division. These events are themselves under genetic control. So phases of cell cycle. Coming to the phases of cell cycle, we have mainly cell cycle is divided into two phases. One is interphase. This is interphase. Interphase consists of G1, S, G2. And we have the M phase. M phase consists of. So coming to the a typical eukaryotic cell will be illustrated by human cell in culture. So these cells divide once in approximately for every 24 hours. For human cell, it takes 24 hours to divide. So the duration of cell cycle can vary from organism to organism. Organism to organism. So and also from cell type. For example, yeast for example will take through the cell cycle in only just about 90 minutes. So the cell cycle is basically divided into two parts. One is interphase and other one is M phase. Okay, so we have interphase as well as M phase. Interphase is again divided into G1, S, G2. G1 means gap 1, S means synthetic phase, G2 means gap 2. Gap 1 phase is actually between M phase and S phase, and gap 2 phase is between synthetic phase and M phase. This is the thing we have to remember between this. And also the 95% of the cell cycle will take place by interphase. Only 5% is of M phase. Okay, so the M phase will represent the phase where the actual division or mitosis will occur. So the interphase will represent between two successive M phases. So it is significantly to note that in 24-hour duration of cell cycle of human cell, so only about an hour will take place for, so 23 hours will take place for interphase, and only 1 hour is considered for M phase. So because the interphase will last for more than 95% of the duration of cell cycle. The M phase is again divided into two types. The M phase starts with nuclear division corresponding to the separation of daughter chromosomes, which we call it as karyokinesis, and usually end with division of cytoplasm. We call it as cytokinesis. So the interphase, though called as resting phase, because it's the time during which cell is preparing for division. So they by undergoing cell growth and DNA replication in an orderly manner. So the interphase is again divided into three phases. So we have G1, which is gap 1, synthesis, and gap 2. G1 phase corresponds to the interval between mitosis and the initiation of DNA replication. During G1 phase, the cell is metabolically active and continuously will grow, but do not replicate its DNA. Or S phase, or synthesis phase. Synthesis phase makes the period during which DNA synthesis and replication takes place. During this time, the amount of DNA becomes doubles. If the initial amount of DNA is 2C, then it becomes 4C. However, there is no increase in the number of chromosomes. If the cell has an haploid number of chromosomes at G1, even after S phase also the number of chromosomes remains same. So in an animal cell during the S phase, the DNA replication begins in the nucleus and the centrioles will duplicate in the cytoplasm. So during G2 phase, the proteins are synthesized for the preparation of mitosis. While cell growth will continue. So coming to some cells in the adult animal do not appear to exhibit division, example heart cells, and many other cells will divide only occasionally, as need to replace cells that have lost because of injury or cell death. The cells that doesn't undergo any further division, we call it as that will exit from G1 phase to enter the inactive stage, which we call it as quiescent stage or G0 of the cell cycle. The cells in this stage remain metabolically active but no longer will proliferate unless we called on to do so depending on the requirement of the organism. In animals, the mitotic cell division is only seen in the diploid somatic cells. However, there are few exceptions to this. Haploid cells divide by mitosis, for example, male honeybees. So against this, the plant shows mitotic division in both haploid as well as in diploid cells. From your recollection of examples of alteration in generation in plants, identify the plant species and stages at which mitosis is seen in haploid cells. You have studied the mitosis in an onion root tip cells. If it has 16 chromosomes in each cell, can you tell the number of chromosomes at G1 after S and after M? So all are 16, 16, 16 because the chromosomal number will not change. And also, what will be the DNA content of G1? G1 is 2C and after S it is 4C and at G2 it is 4C. If the content after M phase is 2C. So this 2C is split into 2C and 2C again. So this is regarding the interphase. In this, most importantly, we have to remember the time duration for human cell and yeast. And next thing is about each and every phase, and about G0 phase, and about the content of DNA and the amount of number of chromosomes, content of DNA, and G2 phase mainly concentrate on protein synthesis, which is and still growing, continues, growth will continues. So this is regarding the interphase G1 S G2. Thank you.